My name is Kendall Putnam. I'm a junior at Meredith College, and I'm majoring in chemistry and chemical engineering. This is my project titled Analysis and Determination of Protein Function as Related to Structure. A little bit of background information on the project. It's based on a study that was conducted by Dr. McKay and colleagues, where they examined a protein whose function is currently unknown, and they entered it through a series of online databases to examine the structural and sequential homologues in order to find proteins with high similarities in order to see if they share a function. And what a homologue is, it's the relationship between proteins that share a common ancestral sequence, and pieces of the original sequence were evolutionarily conserved and passed down into the modern day proteins. And proteins that share these conserved sequences will often exhibit similar 3D structures and therefore will have similar functions. And then this just shows that organisms with higher percentage similarities will often behave similarly in different ways. And this translates into proteins having high similarities and exhibiting similar functions. Then on the right, we see that protein family includes all proteins that share a common ancestral sequence, but this gets further broken down into the domain, which specifies function. But what we're examining in this experiment would be the motifs that are shared amongst proteins with similar functions to see if we can determine the function of a protein based off these motifs. The protein we studied in this project is protein 4Q7Q. It's found in Chitinophaga penensis, which is a soil bacterium, and I modeled the protein here using a software called Pymol. And currently it's suspected to be a GDSL family lipase, which is a subclass of ester cleaving enzymes, but this is not confirmed, it's just the suspected function. This is the process outlined in Dr. McKay's study, in which you take a protein sequence from the protein data bank and enter it through a series of online softwares before determining the hypothetical function. If we continued this project in the lab, we would express the protein and purify it before performing a series of tests to confirm whether or not our hypothesized function is correct. These were the programs used to test the protein 4Q7Q. I'm not going to run through each of these, but it's important to note that each software examines a different aspect of the protein and compares it to the data bank. Then these were the results. This bottom photo is from the BLAST software. And as you can see, this column displays the percent similarity of 4Q7Q to each of these different proteins. And these are really high percentages. And what that tells us is that these are most likely just variations of the same protein in different organisms. So this doesn't actually tell us very much, but it is interesting to note that the top results are listed as GDSL hydrolase family proteins, or more specifically, a GDSL like lipase/acyl hydrolase. Then this top image is from Pfom, and it looks at the evolutionarily conserved positions of specific amino acids. And it tells us that it most likely belongs to the SGNH hydrolase type esterase domain, so cleaving ester bonds. And it's worth mentioning that it was described as having a similar 3D fold to flavoproteins, which are involved in oxidation reactions, also known as oxidoreductases. These were the results from the Moltimate program, which examines the active site of 4Q7Q and compares it to proteins with similar active site positions. And it rates these matches with the RMSD score here. And the lower the score is, the better the match. So these were the two best matches found for 4Q7Q. Enzyme classes 3.1.1.47 and enzyme class 3.1.1. This first one is the enzyme class of platelet activating acetyl-hydrolases, and this one is the enzyme class of acetylesterases. So it's also worth mentioning that esterases are simply a more specific form of a hydrolase, because a hydrolase just specifies that it's an enzyme that breaks a chemical bond during reaction, and an esterase specifically breaks down an ester bond during chemical reaction. And then I didn't put it in the slides, but the third highest match was enzyme class 3.4.16.5, and that's a carboxypeptidase enzyme class. These were the results from Dali and PyRX. So Dali gives us both an RMSD score and a percent identity score. And based off of these two numbers, I determined the top 4 structural alignments for 4Q7Q. 3 of the top 4 structural alignments were acetylxylene esterases, and you can see that makes up a lot of the results in just the top 44 matches. And acetylxylene esterases are defined as accessory enzymes for removing terminal acetate residues from xylene polymers. And they're described as having considerable activities towards substrates like p-nitrophenyl acetate and other ligands belonging to the oxidoreductase enzyme class. The fourth highest structural alignment for 4Q7Q was listed as being a GDSL family lipolytic protein. And again, lipases are a subclass of esterases. For PyRX, this image shows the active site of 4Q7Q in the red region. So this site allows us to take ligands from the enzyme classes specified in the Moltimate program and model them to see which ones will bind to the active region of the protein. And it was found that enzyme classes of 3.1.1.47, which was the platelet activating acetyl-hydrolase enzyme class, did not possess any ligands that would bind to the active site, which was very surprising. So that was removed as a possibility because the binding interactions are arguably the most important part of protein structure. However, when I tested the acetylesterase and carboxypeptidase enzyme classes, they each possessed several ligands that would bind very well in this region. So those were confirmed as strong possibilities. There weren't a lot of ligands found in the enzyme classes specified by Moltimate that would bind well here. So I looked outside of those results and I found that p-nitrophenyl acetate and other oxidoreductase ligands would also bind very well in the protein's active site. So what conclusions can be drawn? The most probable function is an acetylxylene esterase, simply because most of the softwares showed that as a strong possibility, either listing it by name or including it in a generalized grouping. However, we can't fully confirm that. So other strong possibilities would include a carboxypeptidase, oxidoreductase, or acyl hydrolase. The results were not entirely conclusive, simply because some of the programs would give slightly conflicting results, like the Moltimate program specifying that the platelet activating acetyl hydrolase enzyme class being the best match for the protein's active sites, and yet no ligands from that enzyme class would actually bind to the active site. So further modeling would be required before we would ever test this in the lab. And one way that we could do that would be to use the Sprite software, which allows us to compare the 3D structures of side chains in the protein structure to see if they align or not. So this concludes my presentation, and I would like to thank you for listening.